Hi, good morning, everyone. I'm excited to be your host for today's webinar. I'm Deepak Pillai. I manage the South India business for Revvity Signals, and I specialize in financial consulting for software solutions. Thank you all for joining us as we delve into two fascinating topics, each focusing on how digital solutions are transforming R&D in the pharmaceutical industry. I would be introducing our speakers in a while, but before we dive into the agenda, let me cover a few housekeeping rules to ensure a smooth experience for everyone. Please keep your microphones muted during the session to avoid background noise. Feel free to post your questions in the Q&A box at any time during the presentations. We will address them during this dedicated Q&A session at the end. Today's webinar will be recorded and we will share the recording with you afterwards. Lastly, we encourage your active participation in the polls to make the session more engaging. We have a packed agenda today, starting with a poll to understand our audience better, followed by two expert-led talks. The topics are transitioning from paper to digital with a special focus on formulation R&D and empowering biosimilar R&D using our Signals research platform. We will be concluding with a Q&A session where we will address your questions. So let's get started. To begin with, let's take a quick poll to understand our audience mix. The poll question will appear on your screen now. Please take a moment to respond. We will have around 1.5 minutes time to respond to it. So which of the following best describes your main laboratory discipline? Okay, so thank you all for participating. It's wonderful to have such a diverse group of professionals joining us today. So let's move to our first topic. So our first topic is transitioning from paper to digital, streamlining formulation R&D with Revvity Signal software. As formulation R&D evolves, the transition from paper-based processes to digital solutions is essential for innovation and efficiency. This session will cover how Revvity Signals software addresses challenges like data management and user adoption. Our speaker for this topic is Dr. Manish Kandagle. Dr. Manish is a senior application scientist at Revvity Signals. With extensive experience in pharmaceutical R&D, analytical instruments, and scientific software, he brings a wealth of knowledge to today's discussion. His expertise lies in empowering scientists to make data-driven decisions, and his insights are sure to be invaluable as we explore how Revvity Signals can transform formulation R&D. Please join me in welcoming Dr. Manish M. Kandagle. Over to you, Dr. Kandagle.
Good morning, everyone, and thank you for joining us today. In our day-to-day life, we are surrounded by a variety of formulations from medicines and cosmetics to paints, even the lubricants, fragrances, and even the battery materials. We know that behind all these products lies a complex formulations R&D and a process that involves a wide range of operations with various teams and key stakeholders that collaborates across multiple disciplines. And data gets generated from diverse sources such as lab notebooks and various instruments, inventory, that results in a highly heterogeneous data environment with a massive amount of complex data gets generated, and particularly in traditional paper-based systems, where in addition to lab notebooks, various paper-based forms such as sample forms, test requests, COAs, inventory logs, and various physical sign-off documents are used. It poses significant challenges in search and analysis of the right information, and which is quite critical in decision making. It is becoming a major bottleneck in the race to reach the market first, and with increasing productivity demands and stringent compliance requirements, companies are transitioning to a digital platform. At Revvity, we partner with a wide range of customers across various R&D segments. Every day, we collaborate closely with scientists, lab heads, and IT, and many other stakeholders to understand their workflows, expectations from the digital system. And with this experience, we understand the uh, ease of use, data security, uh, integration with the legacy system, and initial investment costs are some of the key concerns, or you can say key expectations they express when transitioning from paper-based or a hybrid system to a digital platform. To address these demands, Revvity Signals Research Suite offers unified cloud-native, completely a SaaS R&D platform that streamlines R&D workflows. It streamlines scientific complexities, enhances collaboration. At the core of this suite is a Signals notebook, which serves as a uh, foundational layer for uh, capturing and managing all the scientific data. All information is securely stored and indexed in a central repository, and that ensures that it is easily accessible across the teams. And the data is stored and indexed in a central repository, and with Spotify pod advanced analytics, you can perform Google like a search, query all the required information, and visualize, analyze via interactive dashboards, which helps in making faster and smarter data-driven decisions. Now let's move to the live system to see how it can help scientists with ease of data capture, collaboration, and data accessibility. Uh, I will walk you through a workflow right from formulation planning to a formulation experimental execution, sample creation and task management to uh, analytical testing and results analysis. Here, while I'm using a pharmaceutical formulation example, it's important to note that the platform works in a similar way for other formulations or any other scientific disciplines as well. Okay, so let's move to the live system, and this is a cloud native electronic system. So what that means, you don't need any installation. You just need to have a web accessible device where you can enter a URL and your credentials, and you can start using the system. So let me launch my Signal student using this link. So it is opened in my browser where I can enter my credentials and get inside the system. Once you enter in your credentials, first thing you see is a dashboard view, and it gives you a high level overview of all the activities, all different kind of data that is being generated and stored inside this system. And these dashboards can be configured at user level. So depending on your roles and privileges, whether it's a bench scientist or a manager or a lab head, they can have different data needs, and as per the data needs and privileges, this data can be populated here. So as a scientist, you'd like to see all different notebooks that you have, different experiments being performed, all recent activities. If you're working on multiple projects with the different team members, then you can track the progress of each of these projects, and you can see everything is hyperlinked. So what that means, you can launch it from here. With a one click, you can open that particular experiment and easily access that information. You can also look at all different samples that are being created, status of the task request, if there are any equipments coming out of calibration, and if there are any experiments or documents awaiting your review. It has embedded inventory capabilities. So if you have ordered any material, you can see the status of those orders as well from here. For example, if I click here, it will show me the status of this order, and it's methanol which was ordered, and it shows that material has already been received. And uh, let's look at some other material. If you click here, this one API which I need for my formulation, and I can see the details of this one, the supplier from where it has been ordered, and it shows me the status which has been received. You can also put a new order from here. So you can go back here, order, and you can enter search for the required materials by entering CAS number or chemical structure. For example, let's say this is a 1027 and 67 2882. This is the CAS number of the material that I need for my formulations experiment. And you enter this, and with the KMSCX database in the backend, which has more than 25 million records, compounds with the different vendors information. So it's query that database and it bring that information here. So it shows me all different vendors that supply these materials. So you can choose uh, based on the price, pack size, and this information. You can select the material that you need. With one click, it can fill in the information into the order form where you can select where you would like to place it as its active compounds, or in which location you would like to get this material. For example, I would like to get it in my uh, cold storage at particular site, and then due date expected for which project you need, and you can fill in all this information. This is how you can search for required material and create a order request form. Many times you would like to see if this material already available in-house with some other teams. So you can query inventory and see if it is available internally, and it shows me that multiple containers are available for this particular material, and where I can see all these containers are available, how much quantity is left, their location, all this information I can see. For example, container 106, it has more than 4.8 kg of the material left, and I can select this material. So with this, I can select this material, and you can create an internal request for this material. This is how you can, in the planning phase, you can check for the materials availability or put a request for new material. Okay. Once you have ensured you have all the materials, I'm discarding it for now. And once you have ensured you have all the required materials, next thing is to check required documentation. So you can also manage all required control documents that are required in the beginning of the in the planning stage. For example, SOPs, material specifications, or control procedures. So you can manage all these documents here. For example, in this case, specific SOPs that you need to perform that particular experiment. So you can upload these SOPs, or you can even create these control documents within the system. So for control documents, just like a DMS system, what you need is ability to edit these documents, have distribution workflows, and roles and privileges and controls. So system allows you to configure this distribution and review approval workflows. So you can have the PDF documents, or you can check the specifications, and all these documents you can have it here, which can be easily distributed, review approved, and shared with the users. Okay. So once you have ensured that all required materials and control documents that are in place, let's assume you would like to start with experiment execution. And for that, user can easily create a new experiment using a template. So by clicking add new, you can select experiment, and it allows you to create a new experiment using predefined templates. So in this case, since I'm in a formulation, it gives me suggestion for the formulations experiment template, but you can also select from multiple templates that you are allowed to access. It will be defined by admin. So you can select formulation experiment template, enter a description of the experiment. Let's say optimization. Select the notebook in which you would like to record this experiment type, the department you belong. You can use this drop down list and then associate with the project. So in this case, when it's paper-based system, in traditional paper-based system, when you create a new experiment, you need to write all this metadata in index as well as on the top of the experiment, and have to fill multiple registers. So here in this case, all this metadata is interlinked. It helps you easily uh, accessing all this information. With then once you fill all these metadata fields, you can create a new experiment. So as it is using a predefined template, it brings the skeleton, the framework for this particular experiment. So every time you create experiment using this template, it brings the consistency in a way you write the experiment. So as you can see, it shows first thing the notebook name. So in traditional paper-based system, since there is no interconnectivity between the data, often naming sequences are quite critical. It gives a long sequences of suffix and prefix, so that just by looking at the name you can track where the data coming from, what is the source of data and where it is going. So whereas in electronic system, you can see this is my notebook. Assuming you can create similar naming sequence, you can configure, you can define that naming sequence in a backend. So here it shows me the experiment number, which is FRD139, and its parent, which is lab notebook MK007. And in this notebook, I can see all different kind of experiments that have been performed so far. And I can scroll through this experiment through review and next. Okay. And then you can see the required metadata for this particular experiment, all the structure of this experiments, which you can see it here. Okay. Another way to create a experiment is either you can create new experiment by template, or another way is to use a existing experiment. If you are performing some experiments repeatedly, you can clone existing experiment to create a new one. So for example, in this case, you can add some of the current experiments to favorites, and then you can make copy a clone of that particular experiment. So let's say this is the one experiment which these steps are routinely used. So you can create this experiment by copy, where it will allow you to create exact copy of this experiment. And if you select copy partial, it will allow you to select the items that you would like to take in a next experiment. So in this case, I'm going to, in interest of time, I'm going to use one of the completed experiment, but of course, I will show you how you can add different materials, or create your formulations recipes. So I'll show you that in live using one of the completed experiment. Okay, so let's go to this one of the completed experiment, and where you can see on the left side, you can see all different kind of experiment contents of this particular experiment. It can be a one experiment, or typically you record perform these experiments in multiple steps as multiple experiments, and you would like to interlink these experiments. So in this case, you can see different experiment contents, whether it's a text, tables, or different materials, libraries, or you can upload different kind of PDF Word documents. In addition to this template, if you want to add additional contents, you can add Excel, PowerPoints, and then Spotfire analysis files. You can upload different kind of reports from here. So in the text field, where you can see it allows you to it gives you the free text field. So you can say, you can type the free text, or you can even templatize this one. So let's say if you can use predefined templates. For example, I would like to enter the aim of my formulations experiment. I can select this and insert this template, and that's how easily you can enter this, fill these text fields. It can save your time. You can interlink these experiments. So preceded by and succeeded by. That's how you can add hyperlinks. So you can add hyperlinks, or you can also connect with preceding and succeeding experiments using this related links. So where you can define, you can establish relationship between different experimental items or different experiments. So in this case, let's say the starting material of this experiment is a product of some other experiment, and that's how you can create a link between these two experiments by preceded or succeeded by. Okay. Once the aim objective is entered, you would like to record the different equipments, documents, and materials that are used for this particular experiment. So you can record different equipments here. In this case, balance, ball machine, and FBD. These different kind of equipments are used to perform this particular formulations experiment, and you can easily add these new materials by clicking this search for the materials. So all the materials, different kind of equipments or supplies, excipients, active materials, raw materials, reagents, everything is managed as a libraries. So if you see it here, it system allows you to manage all these materials using uh, libraries which you can query as and when required, and you can use it. So let's go back to the experiment and see how you can query required equipments and add it here. So from here you can go back here and check for the uh, equipments that are required. So here it shows all different kind of equipments you need, and you can scroll it down, or um, you can select. Let's say I need a colloidal meal, or I need uh, uh, this particular mixing vessel and this uh, balance, and you can select multiple equipments from here, and you can add these selected equipments. So it will fetch all the details about those equipments. So you can see their model number, detail, their operations manual, and the location information. So you can fetch all this information here, and you can also refer the electronic user manuals right inside the system. Once you have recorded equipments, then the next important part is reference documents. Let's say control procedures or standard operating procedures, SOPs, that you would like to refer. In this case, SOP for formulations development is referred here. And similar to equipments, you can query the reference materials library. So it with a predefined filter, it ensures that always you will see the approved and latest version of these documents. That's how it allows you to managing version controls. So you can refer the required documents. Let's say I need to refer these controls of CUs and screens SOP. You can add this SOP, and it brings this information about these SOPs. Then it comes to the one of the most critical part of the formulations development is bill of material, recording the composition details, and the different ways of recording this materials information. Particularly in paper-based system, when we talk to customers, we have seen they manage bill of materials as a separate data sheets, which where they manually write uh, materials that are required, their quantities, and the calculations about the planned material and actual amount. Everything is performed manually, and then it goes to the review approval, and then of course they can perform the experiments. So how you can see it in a digital platform? So here you can see the you can have the admin defined table where you don't have to manually uh, write these compositions. So you can have this material information here. So all different materials that I need to perform this particular formulation with their um, uh, trade names and role in this formulation, batch information, supply, and even you can fetch information about their price, and then you can see the calculations also. So let's see how we can add this information. For that, I'm going to create a new formulations composition right from the scratch, just to show how we can populate all this information. Okay, so for that, you can have a library of admin defined tables where you can query as per your requirement. Let's say formulations, and you can see you can define different kind of formulation composition tables and select the required one, and then you can move as per the requirements. Once it is moved, you can specify the strength of the formulations, what is the mass per unit mass planned, and then batch mass planned, and this will be used for the calculations. Okay. Once it is entered, you can add these materials by querying the respective materials excipients library, or you can scan the barcode or manually enter those barcodes here. Moment you enter the barcode, it fetch all the information from inventory. It's a material ID, and it's a safety and chemical name, all this information, as well as batch number, supplier detail, even price details it can bring. Then you can enter rest of the material details, and with one click it bring all this information. Then you can define role of this respective materials. Checks and binder, so you can update this material roles list in a backend. Filler and this lubricant. So this is how you can define role of the materials. The next important part in formulations composition table or bill of material is calculations. With built-in calculation formulas, it allows you to uh, calculate a theoretically required amount, as well as it calculates percent difference. Let's say the planned amount is for this API is 500 gram, and it calculates weight by weight percent, and then actually weight was something around 4.99.7, and it calculates the difference. So you can enter planned amounts and how much actually weight, and with built-in calculations, it calculates it. You can download it to CSV, and even you can copy these calculations into the CSV format, or in Excel you can calculate it like this. You can also record formulation variations using variations table. So you can add variation tables from the variations tables library. Here I have already added one variations table where you can see it has components, variants, and summary tables. In component tables, to define the master recipe, all the materials that you would like to use for these variations, and then in the variations part, when you create add new variant, it brings the selected materials from the master recipe. So as you can see it here, you can create multiple variations, and you can add new variant by clicking add new variant. It generates a new variation where it brings the list of materials, and then you can uh, you can specify the required quantity of the materials, and that's how it can create here. In this case, it will create 10th variation of this particular formulation, and you can also see all these variations together as a horizontal or vertical summary, where in horizontal summary you can select the information that you would like to see in a summary view. So in this case, there are different formulation trials along with their compositions can be seen as a summary, and same as in a vertical summary, you can see all this information. Often in traditional paper-based systems, the compositions and bill of material is captured in predefined paper-based templates, and similar kind of templates can also be configured. Just an example for that, you can create a worksheet templates that. So this is an example which mimics a paper-based bill of material, where in principle it is same. You can have your text and text elements, product details. You can select this. You can apply as is and water compensation calculations for the active materials, and then edit formulation composition by same as what we discussed in previous tables. Similarly, you can enter barcode or query materials libraries and add these materials here. At the end, you can set up a review approval workflow for this bill of materials. So once it is approved, formulation scientists can proceed with further experiment. Apart from formulations composition, there are many unit operations or processes that formulation scientist needs to record. So you can create a protocol where you can record step-by-step processes that are involved in formulations development. You can have the check marks, date and time stamps, and verifications you can have, or some process templates you can use. So for example, if it is a uh, granulation parameters. So tables can be configured where you can have the top-down list of the respective parameters and values. You can user can easily update this process parameters, or even a worksheet templates like this can be created where you can select a machine, make, and model type, its parameters, shifting parameters, where again the materials that are dispensed can be part of this worksheet, and you can have uh, as you move to this fields, it gives a clue about the data field, what information that user is supposed to fill in this place. You can have the built-in calculation formulas, or you can even calculate the duration based on start and end time and yield calculation. So that's how you can configure different kind of templates. Let's say that after swifting granulation at the end of the process, formulation scientist comes to the compression and coating step. So they can again go and query a worksheet for compression and coating, and from here you can add this template and then fill in this information, type of machine, and the RPM, and that's how they can have the dependent cells. So once you select tooling, automatically it can suggest the dimensions, and that's how you can uh, uh, easily fill this information, do the calculation of the yield based on built-in calculation formulas. Formulation scientists often perform sort of physical analysis of the in-process samples, and you can you can have the pre-configured tables, or you can also continue working with the Excel sheet with Office 365 support. In Cygnus Notebook, you can create Excel templates where you can record all your findings and do the calculation similar to MS Office Excel. So you don't have to go outside. You can continue using Excel within the system. In addition, formulation scientists can take images, do annotations. Once the formulation is prepared throughout the process, formulation scientists uh, uh, create a different kind of samples. For example, in this case, it's a tablet formulation, so they uh, blend sample, mixing samples, or coated, pre-coated tablets, different kind of bulk samples they create for analysis purpose. So they can easily create samples by clicking here, create, click sample, and then they can select what kind of sample. So different kind of sample forms can be created, and formulation scientists can select that formulations sample form so that they can provide contextual information to the analyst. And for example, trial and trial and something like this. Okay, and then they can create a sample, and once the sample is created, that appears in a gets updated into the samples table. So as you can see here, already there are different kind of samples have been created, and for each of the samples you can see the contextual information, what when it was created, its ID and status. So once sample is created, either formulation scientists can preserve it for further use, for that they can register and add container and put it into inventory. So they can inventorize these samples for future use. And also, once the sample is created, they can create a analysis request for these samples. So in this case, this sample is just now created, and then for the sample, function scientists can create a task. So different kind of analysis request forms can be configured. Let's say in this case, analytical testing is required, and then by what time it is required. Let's say it is required by 28 6 p.m. So you can select required by time as well, and assign it to group. So it's ARD. So you can assign it to a group, or even you can assign to individual analyst. And once it is assigned, you select the test for which you are submitting. Let's say submit it for dissolution testing, and you can create this sample. So once the task is created, it shows the task information, automatically creates the task ID number, request link provides the information about source experiment, so that when analyst receives the sample, gets context of this sample. Once the task is created, formulation scientists see the status of this task. Says it is submitted, okay, and then afterwards, either R&D head or ADL scientists can go back to their dashboard, in the task dashboard, and they can see the task information. So once the task is created, this task information gets updated here, where formulation scientist Manish K sees the status of the task, and it shows that status it is submitted, but analyst has not been assigned yet. So next step, uh, analytical R&D manager can assign this task to particular analytical chemist, and then analytical chemist will perform that experiment and send results back to the formulation scientist. So for that, I will log in as a analyst. So Harsha is analytical scientist, and and as an Manish, who is a formulation scientist as well as R&D manager. So Manish assigns this task to Harsha. So assign this task and select a particular user. So in this case, Manish assigns this task to Harsha. Once it is assigned to Harsha, this will get updated into a task dashboard, analyst name, and at the same time, Harsha as an analyst will get a notification. So Harsha analyst gets updated here, and as an analyst, Harsha's tenant, Harsha gets notification that Manish has assigned this task. So Harsha can go back and check this task. Okay. Similarly, she can go back and check in her task dashboard and pick this task and create an analytical experiment for this particular task. Again, similar to formulations experiment, Harsha can pick the required template for this analytical testing. So this is general template, or you can have the specialist templates like dissolution testing, analytical testing, or something for HPLC analysis. So different kind of templates can be created for analytical R&D. So in analytical testing, this template, and then select the notebook in which she would like to record this experiment, the project details. So Harsha will select all these required details and create an experiment. So since the template is selected, it brings that experiment framework for the analytical experiment, which includes the materials and equipments that are used, and different protocols and SOPs, the sample information, and most importantly, the task to do. So task that has been assigned. Here, Harsha can perform this analysis. Let's say Harsha, if she'll unassign this task, and let me go to one of the completed experiment where I will show you how different kind of analytical experiments can be recorded, and how the task can be reassigned. Okay, so let's assume Harsha would like to record this particular task in ongoing analytical experiment. So where she has already recorded these SOPs and reagents that are required, their safety informations, the columns that are required for HPLC analysis. So in this particular analytical experiment, Harsha has performed identifications, IR identifications, where Harsha has uploaded IR spectrum. So you can upload different kind of analytical instrument reports, whether it's in PDF, CSV, or native file formats. You can upload those files with the file upload option. You can upload these reports. Then you can create configurations for HPLC analysis, where Harsha has performed assay by HPLC, and with this worksheet template, Harsha can easily enter the information. So it in this case, it gives an indication of the information that is required, and then Harsha can fill in this information as she proceed with this experiment, enter chromatographic system details, search columns library to add required HPLC column. In this case, in its CODS 3 column has been used. Elution program, system suitability, all this information can be recorded. Then Harsha uploads these chromatograms or perform related substances analysis with admin defined tables, where the calculation formulas have been defined, and upload these images. So the dissolution experiment, so really the template can be created for a dissolution experiment. So with one go, we can go and append template. Let's say it's a dissolution template, dissolution testing, and it can bring entire template for dissolution testing. So that you can see all the content of this particular experiment, dissolution parameters, instrument parameters, which are already recorded here. So you can select apparatus type, dissolution medium, temperatures, as well as detection parameters for HPLC. So you can user can easily record all these experimental parameters, as well as perform calculations for let's say percent release calculation in this case, and attach HPLC reports. So in this case, these two tasks have already been performed, and Harsha can add one more task in this ongoing experiment. So the task which is just now created, so Harsha can add this task into the same experiment. And let's assume Harsha has performed this analysis. Once the analysis is performed, Harsha can attach reports for formulation scientist sample creators reference. So she can go back here and file and upload, attach, let's say, this dissolution chromatogram for sample creators reference. So that's how you can attach different kind of analytical reports. And once it is completed, Harsha completes this task. Once it is completed, sample creator formulator will get a notification that this task is completed by Harsha. So let's say, so if you see here, Manish gets notification that Harsha has completed the task, and at the same time, before sending it for review, Harsha would like to send an email, informal chat with a sample creator. So with comments option, Harsha can send that Manish, I have completed the task. Please check. That's how different groups and team members can collaborate. They can have the chat like experience, and then Manish gets this notification here that Harsha has sent this chat. Harsha has mentioned you, and Manish can see this and respond. And following that, so Harsha will sign this experiment, add reviewer as a Manish, and inviting a reviewer. So Manish will receive the notification that Harsha has sent a review request, and in this case, Manish gets this notification where he can see this has been sent, and as a reviewer, Manish can either reject and reopen this experiment or accept it. Let's say it is accepted. good, put electronic signature and say accept, and all this activity gets recorded into audit trail. So now with this experiment gets closed, and the formulation scientist, as a formulation scientist, Manish will see the results in a formulations experiment. So where Manish can go back into his task table, where Manish will see the results that are posted by the analytical scientist. So it shows status, it is completed, the analytical experiment reference, and also the attachment where Manish can see the dissolution report. Once it is completed, similar to Harsha, if experiment is completed, Manish can sign off this experiment and go through review approval workflow. And all this information gets captured in audit trail, so you can have complete history of this experiment with a date and time stamp, as well as comparison of previous and current version. And then finally, formulation scientists can generate a report for this experiment, where they can select the items they would like to see in this particular report. For example, compositions, and variations, some process informations that I would like to see in my report, and samples and task information. And that's how you can select what you would like to see in the report, and click print to PDF to generate a report. So that's how you can create a report. That's how we showcased how scientists can easily capture their data, and collaborate easily, right from the planning phase to formulation and designing to experiment execution and analysis. And that's how Signals Research Suite can help you in easy transition from the paper-based system to digital platform. Scientists can have more interactive data. Signals Research Suite provides ease of use and faster adoption of the system, enhances collaboration, takes care of data integrity and compliances, provides various integration possibilities with APIs and lab instrument integrations, as well as bringing data from different legacy systems, and helps in saving initial investment cost, as it is a unified SaaS platform. No hardware cost is required. It reduces IT overhead, and helps in a much faster deployment. So that can help in easier and a faster transition from traditional paper-based system to digital platform. And we are happy to discuss this further with you and partner with you. Thank you very much.
So thank you, Dr. Manish, for that insightful presentation. I see we have received some questions, but as I mentioned earlier, we'll be taking it up in the after the second session. So before we move to the next topic, let's take another quick poll. The question will appear on your screen now. We'll have around 1.5 minutes like earlier. Please take a moment to register your responses. I'll really appreciate that. So which Revvity Signals workflows or features do you find most beneficial or would like to learn more about? Okay, great. So moving on. Our second topic is empowering biosimilar R&D, unleashing the potential of Signals Research Suite. Now, in this session, you will learn how Revvity Signals software transforms biosimilar development by streamlining study design, task management, and data integration. With real-world examples, we will explore how these tools enhance productivity, reduce delays, and improve transparency in biosimilar workflows. Our speaker for this session is Dr. Tanvira Paul, a field application scientist specializing in biology at Revvity Signals. Dr. Paul holds a PhD in biochemistry from VIT Vellore, where she conducted a groundbreaking research on bioactive compounds from mangroves. At Revvity Signals, she leverages her deep technical knowledge to support pre-sales presentations, application setup, and customer training. Her ability to translate complex technical concepts into practical solutions has been instrumental in helping our clients maximize the potential of our tools. Dr. Tanvira, the floor is yours.
Hi and good afternoon, everyone. I am Dr. Tanvira Paul and I'm a field application specialist associated with Revvity Signals. Today, in this webinar, we will explore how Revvity Signals research suite transforms the biosimilar development process. As the biosimilar landscape becomes increasingly complex, leveraging advanced technology is crucial for efficient and successful development. Biosimilars are large and complex molecules that are nearly identical to existing biologic drugs, offering similar safety and effectiveness at a reduced cost. However, developing these molecules is a complicated and a very cumbersome process. It involves handling large amount of data from multiple sources having different kinds of formats. It also involves optimizing processes that requires extensive trials and maintaining consistent quality standards through robust monitoring and meticulous documentation. Additionally, managing large amounts of lab materials and resources and collaborating with various scientific professionals or scientists demands significant time and expense, impacting overall productivity and the timeline of placing the drug in the market. Revvity Signals Research Suite, or the SRS, is designed to streamline such biosimilar development workflow. SRS is comprised of Signals Notebook, Signals Vitro Vivo, and Signals Inventor, which are all SaaS-based softwares helping you to make, test, and decide throughout the research lifecycle. Signals Notebook is a cloud-native zero-installation electronic lab notebook that enables fast and easy documentation of research and facilitates both internal and external collaboration among scientists. Signals Vitro Vivo processes data, assay data of all types and formats, while Signals Inventor provides a dashboard for comparing experimental parameters, enabling data-driven decisions. Both Signals Vitro Vivo and Signals Inventor are powered by TIBCO Spotfire, which is an advanced data visualization and analytical tool. For today's webinar, let us consider a typical biosimilar bioprocessing workflow. The process usually begins by creating or selecting a suitable host cell line, and these cells are genetically modified to incorporate the gene encoding the biosimilar protein. Next, in the downside, we have the upstream processings where the cells are cultured and scaled up in bioreactors. Ultimately, the cells are harvested and the expressed proteins is purified in downstream processing steps. Each of these steps involves carrying out numerous or several analyses to determine the quality of the protein or the biosimilar that is being developed. It is very important to also carry forward several assays to find out the stability of the particular biosimilar that is being developed. This ensures that the final product meets the required standards for the safety and the efficacy. So, in order to understand how a scientist will be utilizing this Revvity Signals technology to capture and organize their experiment, now we will migrate to the live system and we will witness how the scientist goes ahead and performs their daily work utilizing Revvity's Signals Research Suite. The scientist begins by accessing their Signals notebook through a web-based browser and entering their credentials, which are end-user specific. After logging in, the scientists are taken to the page where they can navigate to the platform directly for capturing the biosimilar development process. The end user or the scientists can now create several system-defined objects for different kinds of long-term studies and projects. And for this purpose, they go ahead and access the custom objects. In this case, particularly a study design was created to capture the biosimilar development. The scientist now goes ahead and opens the process and checks for the different stages of the workflow. They also go ahead and attach an image. This is made possible by Signals Notebook's image editor, its own image editor, where you can attach any images of any formats, and you can access the informations or refer those images that might be required for the development process of the biosimilars. Later to which the scientist now checks for the work breakdown structure that's give him or her an insight of the different instructions or the steps that are involved in the process. What would be the start date or time? You can also have a checklist for the stages that are being completed, and also register the date and time stamp for the completion of those stages. And you can also now have different analysts who have performed each of these stages in the form of a drop down. Signals Notebook allows you to seamlessly make these table templates to capture the necessary information. Once the scientist checks for the work breakdown structure, now they move ahead and goes forward to assign or generate tasks to assign each of these single steps that are involved in the workflow. For this, Signals Notebook allows you to create tasks of different kinds and types. Once you go ahead and add a content, you can see the tasks under the section. Once you select the tasks, you are presented with different kinds of tasks forms, as you can see over here. All these templates configured in the back end, and based on the types of the study or the experiment that you're carried on, or the kind of the study request you would like to generate, you can go ahead and select that particular template. It also allows you to link different kinds of references, may it be samples, may it be the products, in how many quantities that these tasks are to be created, and also have particular kinds analysts to be assigned for the particular task. In this case, I am self-assigning myself this particular task and creating the task. Once the task is being created, you can see over here, the task can then be submitted for the person to be notified to perform that particular task. As you can see over here, you can monitor the status of the different tasks that are currently being submitted, or that are in progress, or the experiments for these particular tasks have already been created. There are several different new tasks which are still not been assigned or been submitted for a particular analyst to perform. As you can see over here, we can very easily go ahead and submit this particular task. And once this task is submitted, you can see its status change in real time over here. And you can then go ahead and select this particular task and assign analyst based on the number of analysts that you have, or you can even assign this to a group of scientists or your team or the within the organization. Once the tasks have been assigned, the end user will receive notifications in their respective Signals Notebook platform. And these tasks are again linked with the particular request links, the person who has created these tasks. Once the tasks have been assigned and they start performing the experiments, they even get linked with the experiments that link you to the workflow for you to have an access to. It also has different categories and sections, such as what kind of task this is, for you to figure out what particular study type that is being carried out currently. Once the tasks are being assigned, now the next person or the next scientist will go ahead and start their experimentation workflow. The tasks that are being assigned now have a unique identification number or a task ID generated for that particular task. Once you hover on those task IDs, you have access to the informations like what kind of project does it belong to, which workstream it belongs to, when is it required by, who is the analyst associated with that particular task. These task IDs are very helpful for you to connect your experiments that are linked to that particular workflow or the task type, or to which particular study design those tasks belong to. Once the task was assigned, the end user, which in this case is me, was notified within the Signals Notebook platform to go ahead and perform the particular experiment. So we went ahead and created an experiment for that particular tasks, which can be accessed through a particular notebook. As you can see over here, we have migrated to the notebook and selected the first step or the experiment in the biosimilar development, which is the cell line development experiment. Once we enter into the cell line development experiment, we get to check for the task that was assigned. As you can see, since the experiment has already been created, so the task currently shows in progress. Later to which we go ahead and check for the SOP references that might be helpful for performing the experiment. Just like system-defined objects like the study designs and the project, SOPs are also a part of the Signals notebook where you can keep all of your SOPs in a single section. And all you have to do is go ahead and select the necessary protocol that might be used in that particular experiment for the scientist to go ahead and refer to. Next, the scientist goes ahead and checks for all of the necessary materials or the resources that will be utilized during performing of this experiment. The first is the equipments that have been used. Signals Notebook has got its inbuilt materials library and inventory system that allows you to store materials of different types, may it be consumables, equipments, or even biological sequences. Let's just go ahead and see how the scientist feels that there might be an equipment missing. So they simply go ahead and search for the material, which directs the scientist directly to the equipment section. They go ahead and select that particular equipment that was missing from the list and just adds it to the existing list. As you can see over here, how seamlessly the equipment with all of its details, types, manufacturers, models, and the last calibrated dates has been added into the equipment use table. The similar thing was done for adding of the biological sequence, which in this case is the plasmid. As you can see over here, the plasmids and the equipments all have got their unique identification numbers or the IDs that helps you to link that particular resource to your inventory system and all of the informations which are associated with that particular resource. Once after checking all of the important constituents or the resources or the materials that would be used in this experiment, the scientist goes ahead and now adds further sequence that would be important for him to refer. All the scientist has to do is drag and drop that particular sequence within the Signals Notebook platform. As you can see over here, once they drag and drop the biological sequence, because of Signals Notebook's seamless integration with the Snap Gene editing tool, the end user can now freely interact with the biological sequence and can even select sequences that allows the user to copy the sequence in several different formats. And these sequences are also available in the universal GenBank format for you to download in FASTA and the JSON format. Once after confirming this particular sequence, the person or the scientist now goes ahead and checks for the PCR protocols. In this case, Signals Notebook comes with seamless integration with MS Office documents, due to which you can go ahead and drag and drop any sorts of MS Office files. That also allows you to edit those files online or in your system installed MS Office. Once after adding and checking for all the requirements that will be needed for the PCR amplification of the insert, the scientist now scrolls forward and goes ahead to check for the transfection protocol. As you can see over here, Signals Notebook has its own tabular templates where you can go ahead and have calculations associated with them. This helps you not only to avoid any sorts of calculation error, but also helps you to maintain a consistency. As you can see over here, when I go ahead and alter the number, it automatically calculates the amount that would be required. Such these table templates makes capturing of these calculated columns much more easier. Once that is been done, the scientist now goes ahead and uses the worksheet templates that are again can be configured within the Signals Notebook platform for you to capture different experimental protocols. In this case, the transfection protocol, day 3 protocol. uh, the worksheet is available where, as you can see, there are checklists associated, and each of these fields can again be logged once after the information has been fed in. This allows the end user to avoid any sorts of mistake or altering of any of the data once the experiment has been performed and the documentation has been made. The transfection experiment results in numerous petri dish plates with transfected white colonies. Here, the Signals Notebook image editor app was used to annotate the image. And once these annotations are created, we can go ahead and insert particular text that will be again arrested as metadata within the Signals Notebook and are easily searchable. Now, once these petri dish plates were transfected, the scientists will create samples from these plates and register them in the materials library, which will generate unique IDs for traceability back to the plates and plasmids used. These samples, as you can see over here, were generated from the petri plates, and the samples were then directly registered into the materials library section as colonies for us to identify from which petri dish plates these colonies were collected, and these colonies represent to which unique sample IDs. These sample IDs then generated has all of the necessary informations such as which is the plasmid that were used for the transfection experiment, where are the colonies been registered, what is the kind of the colony, when was it created, and what is the particular template for this particular sample. Once the samples are being created, now tasks are further generated with this particular samples for the next person in line to perform the experiment, which in this case is bioprocess by upstream processing for upscaling of these colonies and culturing them in bioreactors. Once after the samples are being created from these particular experiments, as a next step, these tasks were again created for these particular samples, which were again registered as the colonies to perform further investigations. As you can see over here, each of these task information contains the sample details, the date and time stamps. All of these can be achieved again without leaving the Signals Notebook platform, ensuring seamless workflow and collaboration. After receiving the task notification, as we just talked about or mentioned earlier, the next scientist in the line starts the bioprocess experiment for scaling up in the bioreactor. He then migrates to his experiment for the bioprocessing by upstream processing and checks for all of the fermentation conditions, which he can now have templates for, and he can automatically allot or assign informations as all of these are being automatically made available in this particular template. Similarly, in this way, all of the scientists carry forward the experiments within the same platform and even simultaneously. We are aware that the biosimilar developmental process is a very complicated and a complex procedure that involves of several different experiments that are being performed simultaneously by different users, and hence the need for collaboration is very important. Just like we witnessed how the scientists performed the cell line development experiment and optimized the media conditions that are required for scaling up of the cultures, we will go ahead and see how a scientist performs several in vitro and in vivo analysis, and the results that are generated from this analysis can be seamlessly brought into the Signals Notebook platform, and the data generated can be analyzed and processed within the Signals Vitro Vivo environment without having to leave the platform or migrating to a third-party software. Just like the cell line development experiment and the other experiments in this workflow were captured, the scientist now goes ahead and performs the LCMS analysis for a set of samples that were assigned to him. So once the scientists have received all of the samples and the task for performing of the LCMS analysis, he goes ahead and uses one of the most important features for the biologist in the signals notebook is the plate feature. After adding the plate from the add content section, the scientist now has a the entire plate format available to him to go ahead and populate. As you can see, there are different layers that are available to feed in the informations as the substances or the things or the materials that are to be added within the plate. In Signals Notebook, you have the option of adding each of these layers meticulously, and you can even save these plate maps for use of future purposes, because the scientists might need to repeat the experiments twice, thrice, and well, they won't be requiring it to create the plate every time they are creating the experiment. All they have to do is go ahead and select that particular plate map. Before that, we will go ahead and see how we can populate this plate and how by populating this plate format, all of the information gets saved in this tabular template, which is again available to you to be downloaded in the CSV format and can be fed directly into the instrument for the ease of performing of the experiment. Now, the scientist goes ahead and types in the well format that they require. In this case, it is the samples and the standard. All they have to do is go ahead and add to the well format list. Once they do that, you can see you can just select the particular sections and select the standards for them. And you can also select the particular whales for adding the samples. As you can see, it just took me mere few seconds to go ahead and fill in this format. We can also, if we have got more than one plates, we can go ahead and copy the same particular format to the rest of the plates by just mentioning the number of the plates and simply selecting copy. Once the scientist finishes filling in the well formats, then moves into filling informations for the other layers, which are an important component of the experiment. Now they can go ahead and add the concentrations. They can do so by selecting the single column and then copying it to the rest of the columns. They can, and all of this information, as soon as they are being fed, is also being captured along with their units. As you can see over here, you can choose the units and you can add in the concentrations. Similarly, the same informations can again be copied to the rest of the plates if there is any. And now coming to the materials or the samples. Now the materials or the samples were already connected with our tasks lists. So for adding in the informations, as you can see over here, these were the samples that were linked with the tasks and are now plated within the plate for performing of the LCMS analysis. We will go ahead and see how we can add the standards. Now, the standards that were used in the experiments were already present in the material section. So we can go ahead and choose add from the experiment, which will expose us the number of samples or the materials that were used. In this case, the standard that we have used is mentioned over here. So we add it to the materials list. As soon as we add it to the materials list, all we can do is go ahead and populate for that particular standard by selecting this section. So now these samples are directly connected with the samples in the materials library. So you can refer back to the samples or the materials that were plated or to create this particular plate format. Similarly, for creating the amount as well, you have again got the units. You can very quickly fill in the well for the equal amount by simply adding here the number and adding here 10, and you could see that it took me just few fraction of seconds to add this entire format please. Similarly, you can go ahead and have the option of adding several more layers or even deleting or hiding the layers that will not be instrumental or helpful in creating of the plate. Once after the plate is being designed and the format is being fed into the instrumentation, the analysis is being performed. After the performing of the analysis, the LCMS data that was generated in the form of TXT files were simply attached within this particular experiment for it to be processed in the Signals vitro vivo environment. As you can see, the dosage files that will be very important for performing the pharmacokinetic studies is being attached here in the TXT format, and the LCMS analysis data, as you can see over here, was simply attached here, dragged and dropped over here, and it is attached within the Signals notebook platform. The Signals Vitro Vivo provides us with different sets of apps for performing, say, in vivo assays or the screening assays, and these are all validated apps. So in order to process the LCMS data that we have generated and attached into the Signals Notebook platform, we simply use the Data Importer app, which brings in all of the mass spec data. There are parsing instructions that has been provided in the backend for the data from the instrument to match with the way they are being represented in the platform. Similarly, the dosing information data is also being brought into the Vitro Vivo environment for its analysis. As you can see over here, the Signals notebook is connected to this platform, and hence it can bring in all of the features such as the animal IDs or the annotations or the experimental properties. Once you bring in all of these data into this platform, now we simply go ahead and choose the PK parameter app. As you can see over here, you can choose the PK parameter app. Once you choose the PK parameter app, it will call in to upload all of the mass spectrometer data and the dosing data. As soon as you choose this option, it will give you the option of uploading the data. In this case, we'll go ahead and choose the mass spec data that is uploaded over here, quickly matching with the table columns that are present. Once that is being done, it will just go ahead and pull in all of the informations and give you a visualization dashboard, as you can witness over here. Similarly, we also choose in the dosage data. And just like that, we will have all of the informations present. Once we do that, we can go ahead and simply click on calculate the PK parameters. So once all of the informations are present, the app will automatically calculate the PK parameters. The PK parameters will then go ahead and perform the analysis. As you can see over here, you can witness that the system has automatically calculated the different parameters such as the Cmax, the Tmax, the half-life for the drug that was tested or the biosimilar that was being tested. And this is how you can have all of the analysis informations within this platform. Once this information is available, you can go ahead and choose to export these visualizations in the form of the PDF or an Excel, whatever is your option. Once you choose this, you have again option to customize your report or the results from the analysis that was performed within the Vitro Vivo environment. In this case, we'll go ahead and choose all of the pages, which will include all of the app details that have been used along with the mass spec data that were imported from the Signals notebook platform, and how the analysis visualization dashboard is being generated. Once it has been done, you export the PDF format, it exports the report, and now the report is ready for you to attach. As you can see over here, I have simply drag and drop the PDF that we have just generated, and now it is available to you within the Signals notebook platform. This gives the scientists the ease to check the different parameters or the assays that have been performed, and these reports are now readily available to any user who will be performing the LCMS analysis further. Once all the experiments leading to the development of the biosimilars are completed, the biosimilar scientists overlooking the entire process can now visualize all of the outcomes of the different stages in her or his Signals inventor dashboard. As you can see over here, all of the experiments that were being performed during the developmental process of the biosimilars are available in the dashboard, where you can utilize specific filters to compare and harmonize all of the results, which ultimately aids in taking a data-driven decisions. Revvity Signals technology thus helps and contributes to streamlining and accelerating the biosimilar development with efficiency and consistency. Thus, Revvity Signals Research Suite helps in the make, test, decide life cycle of the biosimilar developmental workflow, where it unites all the scientific program work into one platform, spanning from the early research to the developmental phase. Thus, in sum, it can be said that the Signals Research Suite helps to first unify the data from siloed data from all different sources and formats, and is easily searchable in seconds because all of the information that are captured within the Signals Notebook platform are saved as metadata. It also seamlessly promotes collaboration among the chemists and the biologists, which is a crucial and an important step in the development of the biosimilars. And finally, thus helps you to save time and accelerate the entire product development. You can have a taste of the Signals Research Suite by opting for the hyper-trial system, and you can have an access to the system within minutes. With this, I would like to conclude this webinar, and now we can have our question and answer sessions. If you have any, please feel free to post your questions for us to answer. Thank you so much for hearing me out patiently.
Well, thank you, Dr. Tanvira, for that excellent presentation. So before we move to the Q&A, let's conduct one final poll to gather your feedback. The question will appear on your screen now. So which of the following statements about enterprise laboratory informatics systems apply to you? Please take a moment to register your responses. We'll have 1 minute for the same. I would appreciate if all of you would register your responses. Uh, 10 seconds more. Okay, so thank you for for your inputs. So we now move to the Q&A session. If there are people who have not submitted their questions for our speakers, please type them into the Q&A box, and we will address as many as we can within the time we have. So let us move to the first question. So probably, Tanvira, this you may answer. Can we edit biological sequences within SNB?
Thank you, Deepak. That's a very good question. So for biologists, the biological sequences are of utmost importance when we are conducting an experiment. We cannot directly edit the sequences in the Signals notebook, but because of Signals notebook integration with SnapGene, the end user or the scientist is capable of interacting with the biological sequences and can also make a copy of the sequences and the subsequences and download the sequences in the universal GenBank format as FASTA and JSON. I hope that answers your question.
Yes, yes. Okay. I think one more, probably you can handle this. Can templates be made for different workflows, or do we need to make experiments from scratch each time?
So again, the very good question. In Signals Notebook, you are allowed to create templates for each different workflows. And each time a user creates an experiment, they can simply deploy those templates based on the workflows they are using. So no, you don't have to create the experiment from scratch every time. You just need to configure the templates in one time so that it can be used repeatedly as many number of times as you want.
Okay, great. Thanks, Dr. Nandita. Dr. Manish, I think this question would be better. I mean, it will be better if you could answer because I believe it's related to the productivity. So can we get monthly task completed or experiment trends, and and also instruments or equipment logs? Dr. Manish, if you are speaking, I think you are mute. Hello, Dr. Manish, can you hear me?
Yes, now I can hear you. Yeah, I lost you in between.
Yeah, so there's a question for you. Can we get monthly tasks completed or experiments trends? And can we get instruments and equipment logs as well?
Okay, so let's break this in two different questions. So I would like to answer this one by one. The first question was about can we get monthly tasks? So if you can repeat that first part of the question.
Yeah, can we get monthly tasks completed and experiments and trends?
Yeah, I mean, once you create a task, you can have a dashboard view to track the status of this task. So on a monthly basis, you can track what is the status of task and get the notifications. Once the task is completed, you can get the notifications for that. Does that answer that question?
Yes. And the second part of the question was, can we get instruments or equipments log?
Yes, this is again something we'll be happy to discuss with them. So system keeps track of usage of all the equipments, instruments that are used in particular experiment, and the logs can be created. But again, this is something can be further discussed about a specific requirement.
Okay, there's one query related to quality. That is, how do you how do you map editing or correction of errors, and how DQA can review required data and docs?
Okay, so again, every single change which you make in the system gets mapped automatically with date and timestamp, and you can also see compare it with the previous and current version. So and when it comes to the even errors in the writing, so even if it is a typo error, just like MS Office documentation, it shows you the corrections, and every changes that you make gets mapped. Sharing with the DQA, so it has roles and privileges settings can be configured, and with that, the sharing can happen between DQA or any other different departments. Of course, with roles and privileges as per business policies that can be configured.
Okay, thanks, Dr. Manish. There's another question. What data file formats can be uploaded for data processing and analysis? Either of you can take it up.
Tanvira, would you like to take this?
Yes, yes, sure.
Okay, then I'll okay.
Okay, so you can attach all different MS Office files, TXT files, .xls files, and also CSV files can be integrated or attached into the Signals notebook for it to be analyzed further.
Okay, and there's one more question related to data sets only. How does Revvity Signals notebook handle large data sets, and what is its processing power and memory scalability? Okay.
Okay, to answer this simply, Signals Notebook utilizes the cloud native technology for the data storages. So it does not at all bar any kind of memory usage. Scalability is not an issue, and the Signals notebook is capable of handling as much of large data sets as is there and is possible, and it would not affect your performance in any possible ways. I hope that answers. Yes. Thank you.
Yes. Is it possible to create material libraries for the in-house materials? Dr. Manish, would you like to take it up?
Yeah, sure. Yeah, of course. So often in R&Ds, uh, we have a lot of in-house materials, like in-house synthesized materials or formulations, which you would like to use, inventorize and use later on for the next R&D. So this kind of materials, of course, you can create a library of these materials for in-house materials. It can be your formulations or even mixtures. Yes, so answer is yes, of course.
Okay. Then there is another question. It's a long question. Let me put it together. So the question is, a wide range of experiments are conducted in the formulation and biology R&D, okay, each requiring a unique set of data to be captured. How many experiment templates are available out of the box system?
Uh, it's its platform, and so it allows you to uh, configure the templates as per your requirements. So what I showed today was as a concept, but as per your requirements, how you would like to capture your information for each of the experiments, you can have different kind of templates that can be configured easily, and our team can help you. And also, once trained, you can easily create this kind of templates. But system comes as a clean platform. Okay, so that can be configured as per your requirements.
Okay. Final two questions. How to make use of historical data from different types of formulations such as semi-solids or flavor and fragrances? And is there an SAP integration possible?
Okay, so let's break this. I see there are multiple questions again, so we can
Yeah.
go one by one. So the first question which I hear is how to make use of historical data, and quite relevant to the topic of our webinar today. So always like you're transitioning first time from paper-based system to electronic system, we see these challenges, and so in that case, most of the data, if it is in a paper base, always the first approach is to scan those paper-based records and upload it into electronic system. The next step is to do metadata tagging so that all the scanned reports or scanned paper-based copies which you have in the system that can have interconnectivity, and you can search it easily. You can enhance the searchability, and then afterwards you put those scanned copies in structured order, in more in a templatized order. So it brings kind of consistency. So that is a way to move away from paper-based system and making use of your historical data. And then once it is done, then you can start capturing your data electronically. Does that answer the question? So the second
Yes.
question, the second part of your question was, if you repeat it again. So what was after historical data?
Is SAP integration possible?
Okay. So again, system comes with built-in inventory capabilities, but if you still have inventory managed by SAP, we support SAP integration through APIs, or different approaches are there. And of course, this is something which requires our professional services engagement, and that can be again discussed further.
Okay, great. One last question. Do you provide trial licenses? I think this we get almost in all webinars.
Absolutely. And please feel free to visit our website. Sign up for a 15 days free trial. It has predefined templates, so you can play around, get feel of the system. Of course, it has some limitations when it comes to the full-fledged version, but the purpose is to give you the feel of the system. So just visit our website and sign up for the free trial. Okay, and you will get the link, and then you can start using with your credentials.
Okay, great. Thank you, Dr. Manish and Dr. Tanvira. And thank you all for your thoughtful questions. If there are any further queries, please don't hesitate to reach out to us and we'll be happy to assist you. So that brings us to the end of today's webinar. A big thank you to our speakers, Dr. Manish and Dr. Tanvira, for sharing their expertise, and to all of you to taking the time to join us. We hope you find the session informative and engaging. If you would like to learn more about Revvity Signals or have additional questions, please reach out to us directly. Thank you once again, and we look forward to connecting with you in the future webinars. Have a great day.
Thank you very much. Thank you all. Bye.
Thank you. Bye-bye.
Thank you, everyone.